Which one of the following is true about the importance of Lucy in resolving debates about human evolution? Big brains came before bipedalism. Lucy had a brain size of 1.8% of her body mass. Bipedalism came before big brains. Lucy was partially an arboreal primate. First up, question 1 asks about Lucy's importance in human evolution. The correct answer is bipedalism came before big brains. Here is why. Lucy, the famous Australopithecus afarensis skeleton, showed us that our ancestors were walking upright about 3.2 million years ago. Her pelvis and leg bones are key evidence. This was a game changer because it proved that walking on two legs evolved before our brains started getting significantly larger, challenging older ideas. So Lucy helped settle the debate bipedalism first, then the big brain development. Hope that makes sense. Which one of the following can best explain behavior? It is the coordinated response of an organism to an internal or external stimuli, the tendency for parts of plants to grow towards light, the action or reactions of a person or animal in response to external or internal stimuli, a cell or a group of cells that receives and processes stimuli. Moving on to question 2, which one of the following can best explain behavior? The best fit here is choice C, the actions or reactions of a person or animal in response to external or internal stimuli. Thinking of behavior as how any organism, including us, responds to what's happening around it or inside it, is about those observable actions. Option A is a bit too broad, and B is specific to plants, that is phototropism, and D describes sensory receptors, which are part of behavior but not the definition of itself. So C is our most accurate and comprehensive answer. The quick withdrawal of your hand from a hot plate is an example of fixed action pattern, innate behavior, biological clock, key stimulus. Question 3 gives us a scenario. The quick withdrawal of your hand from a hot plate. What kind of behavior is this? The answer is innate behavior. This is a classic reflex. You don't have to learn to pull your hand away from something hot. It's an automatic, built-in response wired into your nervous system for protection. It's not a complex fixed action pattern, nor is it governed by a biological clock or a key stimulus in the way those terms are specifically used. It's a straightforward innate reflex. Which one of the following process describes a biological clock? Removal of one stem from hot object quickly, the growing of plants towards light, circannual migration of birds from temperate to tropical areas, kinesis and taxis of woollies and other simple animals. Alright, question 4 asks us, which one of the following process describes a biological clock? The correct answer is circannual migration of birds from temperate to tropical areas. Biological clocks are internal timers that regulate rhythmic behaviors. Bird migration, which happens early circannual, is a perfect example. Things like pulling your hand from heat are reflexes, plants growing towards light, is phototropism, and simple animal movements like kinesis are not governed by these complex internal clocks in the same way. So the seasonal migration of birds clearly points to a biological clock. Identify learned behavior from the following. Dog trained to hunt, honeybee making honey, child withdraws hand from hot things, ants making nest. Question 5 wants us to identify learned behavior from the following. The answer is A. Dog trained to hunt. Learned behaviors are things we acquire through experience, practice, or training. They are not just instinctive. Training a dog to hunt involves teaching and reinforcing skills. Making honey, a child withdrawn from heat, that's a reflex. And ants building a nest are examples of innate, meaning that genetically programmed behaviors. Only the trained dog shows a clear learned behavior. Which one of the following features is unique to bacteria? Presence of permeable membrane, being unicellular, absence of nuclear membrane, having flagella for locomotion. Let's look at question 6. Which one of the following feature is unique to bacteria? The key unique feature is absence of nuclear membrane. Bacteria are prokaryotes, meaning their genetic material, the DNA, is not enclosed in a nucleus with a membrane. It's just a region called the nucleus. Other options are not unique. Many cells have permeable membranes. Lots of organisms are unicellular, and flagella uh, are found in some eukaryotes too. The lack of a nuclear membrane is a defining trait of bacteria. This item is based on the diagram which shows the different shape of bacteria below. What is the correct name of the shape of bacteria represented by letters A, B, and C respectively? Rod spirochetes, sphere, rod spirochete, flagellated bacille, clusters, spirella, spirochete, sphere, rod. Question 7 is about bacterial shapes. Referring to diagram with letters A, B, and C, the correct sequence of name is D. Spirochetes, sphere, rod. So A represents spirochetes, which are those flexible spiral-shaped bacteria. B represents spherical bacteria, technically called cocci, and C represents rod-shaped bacteria known as bacille. Remembering these basic shapes is fundamental for microbiology. It is possible to produce human insulin in bacterial strain. The insulin gene should be inserted in a plasmid, and the recombinant DNA plasmid should be inserted into a bacteria. What is the role of the bacteria in this process? The source of insulin and modification, the source of plasmid and insulin production, multiplying the recombinant DNA molecule, joining the gene of insulin with gene of plasmid. On to question 8, which deals with producing human insulin in bacteria. What is the role of the bacterium in this process? The answer is multiplying the recombinant DNA molecule. Here is the deal. Scientists insert the human insulin gene into a bacterial plasmid, creating recombinant DNA. When this plasmid is put into a bacterium, the bacterium as it grows and divides makes many copies of this plasmid. It acts like a teeny factory, mass producing the instructions or the recombinant DNA for making insulin. The bacterium is not the source of insulin itself or modifying it, nor is it joining the genes that happens before. Bacteria can cause disease in all of the following mechanisms except changing the genetic structure of host cells, entering living cells and disrupting metabolic systems, releasing toxins as they multiply, invading and growing on the tissues of organs. Question 9 asks, bacteria can cause disease in all of the following mechanisms except the exception is changing the genetic structure of host cell. While bacteria are nasty pathogens and can invade cells, disrupt metabolism, release toxins, and grow on tissues, they generally don't go around altering the host's DNA. That's more the territory of viruses, which can integrate the genetic material into ours. So bacteria cause trouble in many ways, but directly changing our genetic code is not typically one of them. Functional diseases differ from infectious diseases in that they are caused by microorganisms, occur in the brain and heart, are common in females and males, show no signs of damage on the organs. Question 10 explores how functional diseases differ from infectious diseases. The correct answer is D. Show no signs of damage on the organ. Functional diseases are tricky because there is no visible, measurable structural damage to the organ, like you would see with an infection or injury. Think of conditions like irritable bowel syndrome. The organ functions abnormally, but it might look normal. Infectious diseases are caused by microbes. Functional diseases are not limited to specific organs like the brain and heart and don't have a specific gender bias as defining rule. This item is based on the drawing of a bacteriophage given below. In the above diagram of a bacteriophage, DNA and the protein coat are respectively represented by 2 and 3, 1 and 3, 1 and 2, 2 and 1. Question 11 refers to a diagram of a bacteriophage and asks to identify the DNA and protein code. The correct answer is C1 and 2. Looking at a typical bacteriophage structure, the genetic material, which is DNA in this case, is labeled as 1 and is found inside the head. The protective outer layer or the protein coat or capsid is labeled as 2 in number 2, forming the head and the shares structure. So 1 is DNA and 2 is the protein coat. The social impact of AIDS can be mainly revealed by fear of stigmatization, loss of appetite, reduced labor force, loss of body weight. Question 12 asks about the social impact of AIDS. The main one highlighted here is fear of stigmatization. This is a huge issue. The stigma and the discrimination faced by people living with HIV/AIDS can be devastating, leading to social rejection and fear, which can prevent people from seeking testing or treatment. While loss of appetite and weight loss are symptoms and a reduced labor force is an economic impact, the fear of stigma is a profound social burden. Which one of the following is a DNA virus? Herpes complex, HIV, hepatitis C, H1N1. Next up, question 13. Which one of the following is a DNA virus? The answer is herpes complex. Herpes viruses like the ones causing cold sores or genital herpes uses DNA as their genetic material. HIV, hepatitis, H1N1 influenza are examples of RNA virus. So remember, herpes for a common DNA virus. Which one of the following is a correct comparison of lytic and lysogenic cycle of virus multiplication? Lysogenic cycle releases new virus using only chronic cycle. Lytic cycle involves incorporation of viral DNA into the host DNA. Lytic cycle releases new virus only by splitting the host cell. Both cycles damage the host cell DNA following infection. Question 14 compares lytic and lysogenic cycle of virus multiplication. The correct statement is lytic cycle releases new viruses only by splitting the host cell. The lytic cycle is the fast and furious one. The virus replicates rapidly inside the host and then bursts out by lysing or splitting open the host cell, releasing many new viruses. The lysogenic cycle, or mentioned in A and B, involves the viral DNA incorporating into the host DNA and can lie dormant. Both cycles eventually damage the host, but D is too general. In HIV/AIDS community screening, students were asked to take part in voluntary testing. The following discussion presents the views of our grade 12 students named A, B, C, and D toward HIV testing. Student A: I should not take the testing. It's simply boring. Student B: we should take the testing since we are educated. We should be exemplary. Student C: if we get tested and got positive, what should we do? So let us not get tested. Student D: this HIV/AIDS testing is simply a business. We should not be cheated. Which student do you think is correct? Student B, student D, student A, student C. Question 15 presents a discussion about HIV testing among students and asks which student is correct. The best view is from student B, who says we should take the testing since we are educated. We should be exemplary. Voluntary HIV testing is crucial for public health. It helps with early diagnosis, treatments, preventing spread. So student B shows responsibility. Student A is a dismissal. C is fear, and this conspiracy theory are all unhelpful and misinformed viewpoints regarding an important health measure. Which events of cellular respiration takes place in the cytoplasm eukaryotic cell? Production of pyruvate from glucose, reaction of acetyl-CoA with oxaloacetate, production of ATP through chemosmosis, reaction of pyruvate with coenzyme A. Alright, question 16. Which event of cellular respiration takes place in the cytoplasm of eukaryotic cells? The answer is production of pyruvate from glucose. The process is glycolysis, and it is the very first stage where glucose is broken down into pyruvate, and it happens right in the cytoplasm. The other options like the Krebs cycle, B and D involving acetyl-CoA and pyruvate reaction with coenzyme A, and chemosmosis For ATP production, or option C, all occur inside the mitochondria. So glycolysis in the cytoplasm is the one. Suppose two moles of glucose is burned aerobically, how many moles of ATP can be produced by substrate level phosphorylation in Krebs cycle? 4, 6, 10, 8. Question 17 is a calculation. Suppose two molecules of glucose is burned aerobically. How many moles of ATP can be produced by substrate level phosphorylation in Krebs cycle? The answer is 4. Here is how. One molecule of glucose leads to two turns of the Krebs cycle. In each turn of the Krebs cycle, one ATP or GTP, which is equivalent to ATP here, and is made directly by substrate level phosphorylation. So, for one glucose, that is 2 ATP from the Krebs cycle. If we have two molecules of glucose, we will get double that, which is 4 moles of ATP via substrate level phosphorylation in the Krebs cycle. During alcoholic fermentation, carbon dioxide is released when acetaldehyde is reduced, pyruvate is decarboxylated, pyruvate is oxidized, ethanol is oxidized. Question 18 asks us, during alcoholic fermentation, carbon dioxide is released when? The correct point is pyruvate is decarboxylated. In alcoholic fermentation, the pyruvate, or which comes from glucose, first loses a carbon dioxide molecule, that is decarboxylation, to become acetaldehyde. And this release of carbon dioxide is what makes bread rise when yeast ferments. And then the acetaldehyde is reduced to ethanol. So the carbon dioxide release happens when pyruvate is decarboxylated. During anaerobic respiration, reduced NAD is required to split pyruvate to carbon dioxide and acetaldehyde, reduce pyruvate to lactate, converts lactate into pyruvate, reduce pyruvate to acetyl-CoA. For question 19, during anaerobic respiration, reduced NAD is required to, the answer is reduce pyruvate to lactate. In lactic acid fermentation, which is a type of anaerobic respiration, NADH, that is reduced NAD, donates its electron and hydrogen to pyruvate. This regenerates NAD+, so glycolysis can continue, and the pyruvate gets converted into lactate. This happens when your muscles are working hard without enough oxygen. Why do we get tired and fatigued during heavy exercise such as long distance running? It is due to accumulation of carbon dioxide, accumulation of lactic acid, high rate of aerobic respiration, shortage of glucose. Question 20 asks us, why do we get tired and fatigued during heavy exercise such as long distance running? The primary reason is accumulation of lactic acid. When you are exercising intensely, your muscles might not get enough oxygen for aerobic respiration. So they switch to anaerobic respiration, producing lactic acid. The lactic acid builds up, lowers the pH in your muscles, and contributes to that feeling of fatigue and burning. While carbon dioxide accumulation or glucose shortage can play roles in exhaustion generally, lactic acid is key for the specific type of fatigue. If one base in the six triplets of the gene coding for one of the four polypeptides in the hemoglobin molecule alters the triplet from guanine, adenine, guanine to guanine, thymine, guanine, which one of the following biological terms best describes the above phenomenon? Mutation, cloning, translation, transcription. Question 21 describes a change in a DNA triplet from guanine, adenine, guanine to guanine, thymine, guanine in hemoglobin gene. Which biological term best describes this? The answer is mutation. Mutation is any change in the DNA sequence. This is specific change, meaning that from guanine to guanine, adenine, guanine to guanine, thymine, guanine is a classic example of a point mutation that causes sickle cell anemia. Cloning, or option B, is making copies, whereas translation, or option C, is making protein from messenger RNA, and transcription, or option D, is making messenger RNA from DNA, and the change in the DNA itself is a mutation. Which one of the following is the basic characteristics of mutation? It is spontaneous, planned, regular, expected. Question 22 asks us which one of the following is the basic characteristics of mutation. The most fitting answer is spontaneous. Mutations are fundamentally random, spontaneous events. They can happen due to errors during DNA replication or exposure to mutagens, but they are not planned, regular, or necessarily expected in a predictive way. Their spontaneous nature is a key aspect of how genetic variation arises. The theory that suggests life can evolve autonomously from non-living objects is known as Cosmosian theory, spontaneous generation theory, biochemical theory, eternity theory. Question 23 asks about the theory that life can evolve autonomously from non-living objects. The correct term for this is biochemical theory. This theory, also known as abiogenesis, proposes that life arose from inorganic molecules through a series of chemical reactions on early Earth. The Miller-Urey experiment proved some support for this, and Cosmosian theory is about life from space. Spontaneous generation was an old disproven idea of life popping up fully formed, and eternity theory is not a scientific theory of origin. Which one of the following is true about neo-Darwinian theory? There is a natural variation in features and variation which are heritable. All species tend to produce more offspring than possible survival. It takes into account genetics and animal behavior to understand evolution. Development of new feature is important for survival. Moving to question 24, which one of the following is true about neo-Darwinian theory? The best statement here is there is a natural variation in features and variations which are heritable. Neo-Darwinism is essentially Darwin's theory of natural selection combined with modern genetics. It emphasizes that evolution acts on this heritable genetic variation within a population, while B is true or overproduction of offspring, and D can be part of evolution or new features for survival. A captures the core of neo-Darwinism by highlighting heritable variation. C is partially right about genetics, but animal behavior is not its central tenet. Which one of the following statements describes the evolutionary theory of Lamarck? Acquired characteristics cannot be inherited, structures commonly used become more developed, origins of species happens only through natural selection, species start complex and gradually become simple. And our final question 25 asks us which one of the following statements describes the evolutionary theory of Lamarck? The correct answer is structures commonly used become more developed. Lamarck's theory, often called the inheritance of acquired characteristics, proposed that organisms could develop traits during their lifetime through use or disuse. So a giraffe stretching its neck would make it longer. He also thought these acquired traits could be passed to offspring, or which A incorrectly says they cannot be, according to Lamarck. Natural selection is Darwin idea, and D is not part of Lamarck's theory.